Without further ado, I would like to introduce our awesome speaker of this evening. He has over 20 years of experience, during which time he was CIO of Good Start Genomics and has developed data warehousing and machine learning approaches to support the world's largest hedge fund. Please welcome the CIO of Celmatix, Dr. Mark Adams.
Can everybody hear me okay in the back? Yes? Yes? No? Good, all right, I'm getting nods, that's good enough. Thank you for inviting me to speak. And I'm sorry this is a little washed out. These screens look a little better, so if you see slides you can't see here, look up there. What I'm going to talk about today is I'm going to talk about how we can combine genomics and big data approaches to develop personalized medicine. What I'll do is I'll talk a little bit about what both of those things are, what big data and genomics are. I don't have to explain much of the big data side to you guys. I'm going to talk a little bit about how those things have been historically put together and how they're put together now. And I'll wrap up by talking about how we actually did it at Celmatix, and a little bit of like what came out of that. And I hope it'll be interesting. I think it'll be fun. I encourage you to hold questions to the end so I can get through the whole thing. But if there's something like, I don't absolutely understand or that's crazy, I'll answer it, right? But mostly let's just try to get through. And then I will answer questions at the end until everybody's bored of asking questions in public. And then I'll wait around, and if people want to come up and talk to me one-on-one afterwards, that's fine too. This disclosure, you can tell I'm a scientist. I speak at scientific meetings. I'm currently a full-time employee of Celmatix. I'm on the faculty at Bentley University at the Center for Science and Industry, where I run a natural language processing team. And I'm also the entrepreneur resident at Booz Allen Hamilton. I went over the background a bit. I think the key thing here is just saying, look, I've been at this a very long time. I have a Ph.D. in molecular biology. I studied computer science as an undergraduate. I've been doing mixes of those things. Most of my life, mostly doing startup companies in Boston and now here in New York City.
When did you graduate?
1989.
Oh,
I got my teeth.
75.
Let's see.
Couldn't be.
See this gray hair? This gray hair? But I've done a lot of things. I took a detour, did some work in intelligence when I was at Booz Allen. I did some work with hedge funds. My younger brother says that I did the trifecta of the evil college by working for intelligence, a large hedge fund, and doing genetic engineering of human viruses. So I've kind of done it all, I guess. I think this is something you probably all know. The future is data. What do I mean by the future is data? Hope you can see this. In 2010, which is getting to be a long time ago, we passed a very important threshold. We passed the zettabyte threshold of total knowledge of humanity. All the information taken together that we'd ever made ever passed the zettabyte threshold. And for those of you who can't see it, there's a little itty bitty dot you can't even see here, which is 1 terabyte, which used to be considered a lot. Maybe some of you may even remember when that was considered a lot. More interesting than that, in my opinion, is that the digital universe itself has grown, was planned to grow from 2010 to 2020. The estimation was it would grow by a factor of 36. So see this box? This little box is now this box. That means that not only have we generated an enormous amount of information as a species, the rate at which we're doing it is going up really sharply. Now I'll tell you something even more interesting. That projection on digital growth actually fell short of actuality. We passed that point in 2016, so last year. Right, so we passed the expected point in 2020, 2016, so it's growing and growing faster. So, mostly what is all that, right? Well, mostly pictures of cats, right? And this is all the famous internet cats, so my graphics guy did a great job finding internet cats. But it's interesting, a lot of this is just junk. I know junk, it's important to somebody, people like their cats, right?
Data.
This is sequenced DNA, right, as an example. This is what I'm interested in, right? And this is something we started doing relatively recently, sequencing human DNA, right? We did DNA sequencing when I was in grad school. We did it kind of one base of DNA at a time, and it was very slow and time-consuming and really no good. When you look back here from when I was in grad school, right, here we are, then this is basically flat at this scale, and you don't see it at all. This ends at the Human Genome Project, by the way, and that's why we went up at about 3.2 times 10 to the 9th base pairs or so of total sequenced DNA in a human. A lot of the total sequencing we had ever done in the year 2000 was basically a human genome, right? And in fact, it was 2 human genomes, for those of you who are kind of in the know and the skinny. There were 2 racing efforts, one private and one public, to sequence the genome. Somewhat humorously, each of those efforts were racing to sequence the guy in charge. So Craig Venter was being sequenced by the private, and Jim Watson being sequenced by the public. I find that kind of humorous, but hey, it's the nature of things. So that was like 6.4 times 10 to the, whatever, 10 times 10 to the 9th base pairs. But very, very shortly, we had the 1,000 Genomes Project sequencing 1,000 genomes for volunteers. I tried to volunteer, they wouldn't let me, but they sequenced 1,000 genomes. This was quite astounding because it wasn't very long after that we were able to do this. In terms of human genomes, like, hey, as we move forward, now let's fast forward here, we're about here. This is the rate at which DNA has been sequenced, and just some waypoints on how the sequencing was done is sort of not necessarily of interest here. For this group, though, I'll say the interesting thing is the most conservative projection of where we'll go from here today in terms of total human genome sequence, the most conservative is Moore's Law. When have you ever seen a graph of, like, I don't know, the most conservative projection is Moore's Law, where we double every 18 months, right? That's the most conservative projection. The sort of straight line extrapolation from here is like this. It says, and I'm sorry you can't read these here, that means that we will have done the equivalent sequencing of the entire human population at about 2025. It's like, period, everybody, done deal, right? The yellows, I think, where most of us think it'll be, and it's going to peter off a bit too, just because, like many things, we're really talking about sequencing everybody in the developed world at least once, and maybe some portion of the population. But that's what I mean when I say, like, look, this isn't crazy for me to say that every single person here, somewhere in the next few years, will probably have their genome run, either because they choose to do it, or it's just a routine test. And one of the things that's driving this, interestingly, one of the things that's driving the floor on the price of these, invert that graph and say, what does it cost to do a single human genome? Well, the first single human genome costs, I don't know what, $3.5 billion, whatever the Genome Project costs. Pretty quickly, those genomes were running at about $100,000 each. More recently, we're paying about $1,600 to do them, right? This is $1,000 genome people talk about. What's really driving that, by the way, is that medical tests run about $2,000. Like, if you're going to do an MRI series, it's about $2,000. You're going to go do a general medical test, it's about $2,000. That's kind of the floor. They're like, well, we can charge like insurance that much money and they'll pay it, so this is probably where this is going to sell. And the nice thing is you do your genome, you do it once, and you're finished, right? There's some tricky things, and I'm sure those of you who are in this business, if any of you are, recognize that, like, the tricky thing is what you do with that genome, where you keep it, how do you keep it accessible so you can actually use it in the future. But the truth is, people redo MRIs all the time, like at a couple $1,000s. So what does this really mean, right? I like to point out, like, every generation has a signature big science project. Like, people I remember as a kid, like, the moon landings were a really big deal. It was very exciting. We watched it on television. Ours is interesting, as I believe the sort of the big science thing we're doing is the collection and sequencing and analysis of the genomes of a wide range of species. Being able to do so, right, provides really interesting and important insight. I'm not going to talk about that today. I'm going to talk primarily about medicine, because that's sort of the business that I'm in right now. But I'll remind you that we're already today seeing impacts of these big genome sequencing projects on things like improvements in crop yields. We're seeing this with obvious improvements in medication and understanding more about the natural world. And I'm already seeing this in energy. There's a very interesting project that I've worked on with my colleagues at Bentley, looking at how to genetically engineer algae to make and retain oil better, just to make it a better species for making that kind of oil production. So there's a lot of neat, cool things that people are doing, but how does that kind of happen, right? I mean, one of the reasons that I'm here talking to you guys, and one of the reasons I tend to speak about this a lot, is collaboration outside the life sciences has contributed a lot to what we call theoretical biology. So what we used to call what I do, right? Back when I was in grad school doing molecular biology and mostly making, genetically engineered viruses, the idea that you would then go do that computer stuff, back, as I said, sort of in the late 80s and early 90s, people were like, well, that's crazy. One person on my thesis committee told me I was committing career suicide by going into that computer stuff. This was right on the crux of the .com boom, so that was a little funny. But what this says is biologists. Does anybody know who these people are? I mean, just out of curiosity, anybody? Close. This is G.H. Hardy, famous mathematician, famous for being friends with Monjan, probably one of the greatest mathematicians of the early 20th century. So this is Hardy, this is Weinberg. The Hardy-Weinberg equilibrium, which is illustrated graphically over here, was a really important piece of math that biologists use, right? But here's the funny thing about biologists. I taught genetics for a while in genomics at Northeastern University. I used to ask the class, hey, everybody who's got an advanced degree in biology, put up your hand. About half the class would put up their hand. Great. Everybody who's done advanced mathematics, and we'll just make it a low bar, like anybody who's done elementary differential equations or more, put your hand up. All these hands down, all these hands up. Like disjoint set every time I ask that question. Biologists are literally rarely trained as mathematicians, right? Well, Dr. Weinberg and Dr. Hardy played cricket together in Cambridge. Dr. Weinberg went to Dr. Hardy at one point over beer after playing cricket and said, you know, we're really having trouble figuring out what happens to genes in a population as they kind of mix. You know, these mix, they come together, they come apart, how does that work? He explained the problem. Hardy, the mathematician, goes, oh, that's really easy. And he sat down and wrote it out, gave it to Weinberg. Weinberg picks it up and runs out and goes and writes one of the most important papers in population biology, this Hardy-Weinberg equilibrium, that tells you, hey, given a certain number of genes in the genome in the population, like as these people are intermating, at what point do they reach equilibrium? Does it just stay there, right? Hardy, to his death, literally is like, I don't know, like why they call it that. What it's really called is the binomial theorem. And for mathematicians, this is no surprise. Of course you use the binomial theorem. Biologists, not great at math. But what this means is, like, groups like this, right, actually have a disproportionate potential impact in biology. Because as we're doing this, and I'll go into this a bit in a minute, what the genomics program, if you will, like that approach has done is it's converted a lot of what is pretty squishy biology when I was first studying it into hard math, right? And the kind of hard math that it turns out we've gotten pretty good at. So along with genome data, right, things like medical information, personal history, all kinds of those things can significantly improve, health and save lives, literally, right? And I mean, just to give you a concrete example, this is some work we did at Booz Allen where we said, look, we asked a question in severe sepsis and septic shock. Do people know what severe sepsis is? Severe sepsis and septic shock is a bloodborne, usually bacterial disease, often acquired in the hospital. It is lethal for about half the people who get it, and in about 72 hours. Now that sounds really terrible, right? I mean, like you get this disease and you're pretty much going to die in 72 hours. Why this makes a particularly good laboratory is the data that's collected by an individual who doesn't move, that person's not leaving the intensive care unit for the duration of that illness, the whole natural history of that illness is collected in one electronic medical record set. This is very hard to do, for example, for cancer, where like you're going to see a lot of physicians, it's going to take a long time, it's at least 5 years for people to have something meaningful happen. What that means is a disease like this, where you're like, this happens, it's diagnosed, you intervene, they deliver, die, and it's very fast. What we did was, we just said, well, if we pulled and used some natural language processing techniques, I won't go into them here, to pull the data out of the electronic medical records of these patients at what we'd say like a mid-sized hospital system in the Midwest, Sisters of Mercy Health. It was about 400 hospitals and clinics. They had the advantage of being on the same electronic medical record system, right? So we take each of these patients, string their medical history up like beads on a chain. We applied hidden Markov models, right? This is a technique that many of you are familiar with, to say, hey, the ideal treatment, like we know what the treatment should be, looks like this, and we can thread the patient's life histories through that treatment and get a goodness of fit. For people in here who do math or use this, that's actually not a very hard thing to do from a math standpoint. Somewhat surprisingly, nobody had ever done this before. What they found was, if you can look at this graph, looking at the amount of compliance with the, like this is not a secret. Like the hospital had rules, like somebody's got severe sepsis, you need to do this and this and this and this and this. Well, evidence-based medicine, right? The amount of compliance, this is the, we say that the mortality rate, like the likelihood you're gonna die, goes straight down based on the amount of compliance with a correlation coefficient of 0.82. That's like a straight line, right? And it goes, well, if at the lowest level of compliance, 60% are gonna die, and at the highest level of compliance, 20% die. This isn't a new drug, this isn't some crazy breakthrough. It's like, look, we have this rule, if you follow it, people don't die. It's a little surprising that one can take this kind of data, big data, right? I mean, it's not quite petabytes of data, it was a lot. Apply tools like natural language processing and hidden Markov models and all the things you guys already know about, things like sentiment analysis and ad tech, and then find a really striking result like this. And the truth is, this is all over. This was almost the simplest possible problem, but there are many, many more like it, and I'll talk about more in a minute. So, like, this seems really cool, right? Wow, apply math, apply computers, apply technology. How does this all go? So now, strap in, folks. You're going to get the world's shortest and worst biology class. Anybody here is going to go take a biology test afterwards, I apologize, because you won't be good. What's going to follow is the fastest, most incomplete biology lesson ever. If you already know this, I'm sorry, bear with me. If you don't, hold on. Just as a quick point, this is what we're going to care about for the purpose of this. In a patient visit, like go talk to a patient, you remove cells from that patient. That could be, we draw blood at Celmatix. We also have got a spit in a tube. You can do it by buccal swab. Basically anything that gets in not that many cells. In Gattaca, they like do one hair and put it in. That's probably enough too, right? You take the cell, the nucleus is where the chromosomes live. You take the chromosomes out, right? You do this using chemical techniques. It's not terribly complicated. All the genetic material in these chromosomes, again, within those chromosomes, 3.2 times 10 to the 9th base pairs, remember? So big long strings of DNA. A gene is just really a region, it's a segment in the DNA, which is associated with a trait. And the DNA is what makes up those chromosomes, right? So you go from a patient, collect cells, get the nucleus, get the DNA, get the nucleotides, right? Like I said, just the entire plan of view, I mean, this usually is kind of striking to people, like, enough information to completely reproduce you, at least from a biological standpoint, is contained in something about the content of like a DVD. And I don't mean an HD DVD or 4K DVD or like any good kind of DVD, just the old bad kind, is really about the amount of information per cell in you and the complete plan. I find that intriguing. And those, secondly, differ. One DNA sequence to another, one person to another, differs on the average about every 1,000 bases. Remember, there are about 3.2 billion bases, and you differ every 1,000. That's also striking. What's interesting also is that you also differ from most great apes, about 1 in every 1,000. Let that say about your college roommate what you, right? So, take that DNA, right? And remember, the DNA is replicated. It's transcribed into RNA. Remember, hey, everybody knew these acronyms, so I don't need to do it. You transcribe into RNA. RNA is translated into proteins. Remember, that gene is just a region of this that's copied to RNA. That's then made into a protein. That protein undergoes actually a fairly interesting, and we won't dwell on it, protein folding process to yield a three-dimensional structure, right? My postdoc was spent trying to take this string, or no, more properly, this string of just A's, T's, G's, and C's, and predict that shape. I chose this because there's a thing called the Leventhal paradox that basically says that can't be done, period. Not in any thorough way. So I figured it was kind of career insurance. Like I knew we could never get all the way here, so I'd have something to work on in my whole career, right? But it is interesting, right? I mean, a more accurate picture of this is something like this, a space filling model. But remember, these are fully formed operational structures. These genes generally, in most cases, fold into a thing that's a little molecular machine that does work of some kind. Among other things, quite recursively, copying DNA, right? Like everything, all the machinery in your cell is made by and large up of these things, the plan for which is that DVD I was talking about, right? Great, well this is super interesting because now we know kind of, hey, the data in that system makes a thing that we can go then look at biologically and we think about it, right? Great, well how do we tend to a computable form, right? Like how do we really do this? Well, this is an old school sequencing machine. I have to apologize for anybody who knows new sequencing machines don't look like this anymore. But essentially that machine takes a sample of extracted DNA, like remember we took those cells, we extracted the DNA, we do some chemical reactions to it, which I'm not going to detail here. Those chemical reactions essentially allow us to pull the information out, feed it into a computer, and then carry out a number of algorithms to take the sequence, and that's just the A's, T's, G's, and C's, the underlying DNA bases, put them together. And what I'll say for this group is interesting. When I was in grad school, the algorithm that we use for putting these sequences together, remember at the time I was doing this, you'd maybe sequence maybe 100 or 200 bases at a time. So remember that 3.2 times 10 to the 9th bases, you'd look at a couple hundred at a time. You had to put them together, right? Well, when I was doing it, you did it manually. We just literally went and looked at bands on gel, and you wrote it down, and then you went and figured out how to put it together. As we went to sequencing automation, we started to get lots and lots of sequences. Again, somewhat interestingly, little short things that one then had to put back together. So imagine taking 3.2 times 10 to the 9th, or 3 gigabytes of a file, It's just a big one. Maybe it's everything like every author in English ever wrote or something. Chopping it up into 100-character long 20 or 30 or 40 word bits, and then putting it all back together, right? That's actually not impossible to do. I mean, computers are pretty good at this kind of thing. The logical way to do this is you say, well, we'll build a big connected graph. We'll make the edges anything that matches, and the nodes each of those little bits, right? Like, hey, we've got that little bit of text or DNA sequence and this little bit of text, and if they overlap and match, we'll make an edge between those nodes. And the kind of the logical way is like, hey, we'll do that with everything, then we'll just walk the minimum path. Like, this seems to be a rather straightforward graph theory problem. We used to laugh about this when I was a grad student, like, oh, someday, maybe that's how we'll do DNA sequence, right? Nobody had even done a complete genome at the time. Mike Waterman, who was a good friend of my thesis advisor, Tony Smith, Mike Waterman said, someday we'll do this. Like, this was over beers at the faculty club, and I think he thought it was kind of funny. The irony is that today, with the massive amounts of compute and memory that we have, that's actually how we do it now. We build a big connected graph and we span the thing, and then we have a lot of hints because we basically, remember, those genomes only differ once in 1,000. With those hints, we can actually trim that graph and do it in a reasonable amount of time. But it is interesting, like, it's not as complicated as you think. It used to be really complicated because we had to cheat in all directions to be able to make this even work. Now computers are bigger, they're faster, there's basically infinite memory, you can do it. The thing I like to remind you is like music, right? And you can't see this, I'm sorry. This is just big long runs of A's, T's, G's, and C's. And this is like a music staff, right? DNA is structured and layered, right? It's complex. DNA is on one hand just a string of characters, but there's a lot of data encoded in that, right? And I like music because you say, well, music is just fairly complex. There's things encoded in it, right? You listen to a piece of music and it's got rhythm and it's got harmony and it's got melody. And in fact, something I won't talk about here today, but that I've done because it's sort of interesting, is to say, hey, can you translate DNA, the A's, T's, G's, and DNA into usable, listenable music? And it turns out you can do that, and you take advantage of this isomorphism. I'm not going to talk about it today, but it's kind of interesting. For this crowd, though, this might be a little bit more relevant, right? Here's some DNA, like, that's a location. This is real, by the way. There's a location, like 2483959 on its own chromosome. It's just a point in the string. Here are the three characters, ATG. What do ATG do? Well, ATG is the start. It's the start codon. It carries an instruction that says, okay, start translating, then start that protein here. And then here's the next one, 3 over, GCG. Wait, those three bases are an arginine. It's a particular amino acid. Here's instruction to splice the DNA, or to splice the RNA. Here's to end the splice. Go down here to TAA, here's the end of the thing. You know where I'm going with this, right? Like these codons, this is my favorite 6502 assembly language because, yes, I still remember some of the opcodes. Here's 6502 assembly language. Look how familiar it is. There's an address, like that's location. Here's the byte. These just happen to be 2 nibbles, right? Old school, 6502. And here's the instruction opcode that's related to that. And what does this do? This thing taken together prints out A through Z. This thing taken together makes a protein called alcohol dehydrogenase. Right? It's a computer program. It's a computer program, programs and machinery that sell to do something, right? I mean, the data looks more like this. It's terrible. But I mean, data looks like this in ASN1 and it looks like this in XML. It's just, but the point is, it's just data, right? Being able to sort of go from the underlying, we'll say the underlying biological information into data that's computable, and computable in a way that we're all familiar with. These are just strings. They're big. But these days, it isn't that big, right? So the clinical data is the code, right? So we talked a little bit about kind of handling the clinical data with the Sisters of Mercy stuff around serious sepsis. But the truth is, in general, anything we really want to do just can't end with the genome. There's just not enough information there. We try, and there's a lot of interesting things you can do to think about the genome. But really, the best thing you can do is go look at phenotype. Again, sorry about the bad biology lesson. Genotype is what your genes are. Phenotype is kind of what you are. So think about the phenotype as like, well, the genotype programs you, and the phenotype is what comes out. Clinical data can be treated as an important phenotype. I say this as an example. This is from us at Celmatix. One of the things we're doing is collecting outcomes data. Celmatix works, so we're a women's health company. So it's the next generation women's health company. We're particularly interested in women's infertility. That's kind of our main business. what we need is to collect outcomes data. And outcomes data is really, in this case, electronic medical records data. These are just real treatment cycles from about 670,000 of them. Remember how I said how interesting it was to have the whole history of a severe sepsis patient, like from the beginning to the end, so we could work on it? One of the reasons I came to Celmatix to work in that business is that, like that severe sepsis case, infertility is a case. Somebody goes to an infertility, an IVF clinic, an infertility clinic, and in general, they go into the clinic and they stay there for a whole cycle. And by a cycle, I mean, they'll get diagnosed, they'll decide what might be wrong with the person and do an intervention. Then they'll see if that person gets pregnant, and you'll know that within, like a couple of months. Right? This isn't like this impossibly long thing. You may have to do many of those cycles, but for the purpose of this case, one cycle where you do all of those things are almost always in one place and in one electronic medical record. So it means this is cool is we can go, okay, we've got infertility clinics all over the country that we have research agreements with, and we've collected all this data, 670,000, it's more now, it's almost 700,000. 700,000 electronic medical records is really cool and exciting, right? You can do a lot of stuff with that. Downside, unlike the Sisters of Mercy example, these guys aren't on the same electronic medical record system. It would be great if they were, they're not. So we have to do a lot of kind of mix and match and harmonization, and I won't go into too much detail because I think people would be bored and quit. But to say, like, we do a lot of harmonization and standardization of that data. We do natural language processing to take the doctor's notes and kind of tease out what's going on, and then standardize that into a system that we can then manage that. I'll show you a picture of that system in a few minutes. Where does this end up, right? Like, what are we trying to do here? And I mean, what are we trying to do in Celmatix? Like, we're trying to cure infertility. Like, yeah, that's good. But what are we trying to do generally with this? There's something called the learning healthcare system, right? A learning health system basically says, look, you're going to take care of patients. You're going to take care of patients, and you're going to yield data and information that you can go do science on, right? And you'll say, hey, science means we have some ideas or concepts about what we might do. And those experiments can then yield evidence. And this is very much like the severe sepsis show, right? Hey, in severe sepsis, if you do this thing, the evidence suggests that your patients will not die as much, right? And then that evidence can then impact care. And here in the severe sepsis case, that's what happened, right? They said, uh-oh, we did some science, we did an experiment, I showed you the data, uh-oh, that evidence means we need to change the way we're doing it. What this had to yield, what this had on care is that they hired a bunch of nurses and gave them clipboards. I mean, this was not a high-tech solution. Hired a bunch of nurses, gave them clipboards, had a list of that protocol for severe sepsis, septic shock, and as soon as somebody was diagnosed with that, they were assigned a nurse with a clipboard who followed the physicians and the nurses and everybody around and made sure that they followed that rule. And the reason is, like, if they did that, they could see, as you saw, almost 60% decrease in mortality, right? So doing this, now, again, as I mentioned before, this is a goal for lots of things. In oncology, this is a very active area in cancer treatment. People are doing a lot of work on this. But why is it exciting in the fertility space? For the reason I told you before. You can get access to that data. People are highly motivated. It's not like oncology in that sense. People want to have babies. They really do. Like, they'll do things. They'll really work at it. So having this loop be closed because you've got access to the information, it doesn't take very long to get to the endpoint. You can apply big data and algorithms and analytics, and they come up with interventions that people actually do, right? So that's kind of why he says, why are you doing all this? Why are you wasting all this time or spending all the investors' money or whatever? So cool, right? Those are all the bits and pieces. Let's talk about applying. And again, from a background standpoint, as I mentioned, we're a next-gen women's health company. We're really trying to use big data and genomics and all these other advanced next-generation tools to help women make good fertility decisions. Really to get pregnant, right? And just as a reminder, you guys may not know this, like on the order of 12%, it's probably a little higher than this, like have infertility issues. So this isn't small. And they generally don't begin to assess it until it's really late. And I think, again, something probably everybody knows intuitively is important to mention it is that clock is ticking. The number one principal component, if you go to an analogy, that 700,000, like, when you look at it, guess what it says? When you get older, it gets harder to get pregnant. Oh, really? Like, wow, that was exciting. How much you spend to find that out, Mark? Like, yep, we know that. It's all the underlying things that are complicated. And the fact that that clock is running and ticking and getting worse. I'll tell you another thing. We did a survey of millennial women, 5,000 millennial women. And one of the things we discovered, we just asked them, it's like, hey, what are your plans? Like, what are your plans for your family building? And I'm going to get the number wrong. I think it was 82% of these women assumed that they would be starting their family journeys in their mid-30s. Most. to some extent almost all, are like, in my mid to late 30s. It's not a good question. You realize that people who start their fertility journeys that may have problems with this, would you be interested in blah, 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 blah. Many, most, a large plurality, were not only willing to use infertility treatment, they're just assuming, like, oh yeah, I'm just gonna use that again. It's caused a bit of concern. I have millennial children, right? So I'm like, do you think that's a good plan? But that said, the problem here is that you're already aging. If you're coming in late, you're already late. You really need to diagnose this correctly and efficiently and effectively to maximize your chances. And then, of course, $1.5 billion drug market alone is spending $4 billion. There's a lot of room here to make this better. And remember, this money in the US, by and large, except for Massachusetts and a couple of other progressive states, is entirely out of pocket. For those of you who don't know, we're talking like tens of thousands of dollars, right? So there's a lot of money out of people's pocket. There's a lot of efficiency, and we believe some strong ways to make this better, right? So, what do? And we mentioned this before, right? We collect a lot of data, we analyze that data. So I get ahead of this. And then we embarked on annotating what we know, right? And when I say what we know, like I think many of you guys who are in the scientific business, who've been around or studied in grad school, the scientific literature is basically a repository of everything we know, at least everything we know to a very kind of strict sense of peer review and careful replication and everything else. So if you say, well, how can we really figure out what do we know about this? A good way to do it is, well, we'll go to the literature and we'll read it, right? And I think one of the challenges here, and I'm sure everybody here recognizes, the literature isn't very computable. We can't just go plug the computer into the literature and have it tell us the answers, right? These are papers that are written by human beings, for human beings. I thought when I was in grad school and writing papers, sometimes nobody's going to read the damn thing. It's too complicated, not very interesting, right? So what we did was, I'm sorry, you read this, there are about 25,000 peer-reviewed journal articles that you can look in the literature if you put the words female infertility in genes, genomics, genetics, about 25,000 papers. 25,000 papers is a lot of papers for somebody to read. You would be reading papers for a very long time if you read all those papers. We did manually curate those though, right? Because the truth is, and again, I think most of you probably recognize this, you can flip through those papers and pretty quickly, maybe by reading the title and the abstract or the title and where it was done, who did it, whether or not it's likely to be interesting. Right? People can do that. In fact, we did have people do that. There's a sidebar that I won't go into here. One of the great things about doing this once is that now we've got this enormous collection of human curated papers that have turned unstructured data into structured data and made things like deciding whether to read it or not, or what that category is. We've been going back and looking at a number of things, particularly recurrent neural networks, looking at sort of deep learning approaches to say, hey, how good is deep learning and other AI techniques at doing this exact thing? Because we have a lot of examples now, like 25,000 papers worth of examples, right? I'll say as a sidebar, a good neural network, a well-trained recurrent neural network, is about as good as a bad grad student, for what that's worth, right? The grad students don't like to hear that. When manual curation, right? And I mean, by manual curation, just to be clear, we built a formal data model, like one that's in Neo4j and sort of formally managed. We then exposed that formal data model literally as like a web page, an ugly, not very nice web page with some interactivity. And then we had a very large crew of curators do nothing but read these papers and annotate just fill in the web form for the data elements that we agreed we wanted. We had multiple people do it, and then we did, as you might expect, we then looked at where there were potential errors of non-overlap by doing double curation. And we ended up with a subset of, I'd say, you read this here, about 1,700 genes. These 1,700 genes were things that multiple readers from many papers agreed were involved in the infertility process, right? We then ranked them. The advantage of sort of coming behind cancer, remember I said oncology has been way ahead on this? Obviously, motivated population, cancer is a disease of the genome. They've been doing this. We could then say, hey, we're going to borrow their approach. This is called ClinGen. The ClinGen group is the group that sort of defines how do you know that data in the literature shows association between genes and diseases, or genes and outcomes. You can follow this process. And then doing this, we ended up with about 201 gene-disease combinations. Like this gene was related in some way to this disease. Of that, there are about 427 genetic variations. Remember those cases where I said that in, the average genome, about 1,000 bases apart is invariant? I really just mean one base that's different. So it's like, one of these strings that's exactly the same. You hit a base that's different, about 1 in 1,000. About 427 of these variants had an association with the inherent diagnosis combination that was supported by ClinGen. We then did something called meta-analysis of those studies. So we said, okay, I've got all the papers that these things came from. Let's go do meta-analysis. And this just means literally going to the papers and pulling the populations out and piling them up one on the other. We have to do some very careful inclusion and exclusion to make sure they match, make sure they're not the same population being used by 2 different physicians. There's a lot of that stuff. And then you just do simple statistics. You say, okay, what's the p-value that this association is accurate? And we took a very, very strict cutoff with that. And we ended up with about 32 genes, about 49 variants, we're up to about 70 something now, which have absolutely airtight evidence. So this is kind of a partly manual, partly machine process to identify these. But what we ended up with was a very robust set of genes. I'll show you at the end what we did with them, but keep this in mind, that don't lose these genes here, because these genes and these variants turn out to be really interesting. But remember, this isn't anything we discovered, like, oh, Heterogeneity, Celmatix said something really cool. Nope, we just went to the literature and summed up what everybody else knew and boiled it together.
What was the cutoff?
The p-value cutoff? It's complicated because we did it, it's an OR cutoff, but it's equivalent to about a p is less than or equal to 0.46, just about a p05. So I mean, we wouldn't have done it if we couldn't have gone, hey, that's statistically significant, but it wasn't the driver. Because we're really talking about statistics, we're looking at risk. And we can talk about odds adjusted risk ratio all day, I'm sure. I like it, nobody else does. Great, super, Mark. You did all this work. In addition to that work, and I won't talk about it here because we talked so much about DNA sequencing, we're also doing some independent DNA sequencing efforts. So we're doing whole genome sequencing. I like to say we've sort of got the 1,000 Genomes Project of Human Infertility. We've got about 15, almost 1,600 now samples that are in various states of being whole genome sequenced. Remember, these are cases for which we have the complete electronic medical records too. So it's really important to say, hey, we know what was in the literature, that's pretty interesting. Now we want to go look closely at these markers in a more detailed sequence population. Why this matters is these statistical associations, the question that you may not have heard up here in the front, like, hey, what was your p-value cutoff? Like, how was your statistically doing this? The reason for that is that she recognized that the work we were doing here is largely a statistical association of those variants from the literature and outcomes. Those are markers, though, and again, this is sort of inside baseball, and forgive me, like when you do genetics, classical genetics, you're looking at statistical associations between genotypes and phenotypes. Up until very recently, like gene sequencing recently, like since I've been in grad school, you couldn't really go look exactly at the genome and go figure out exactly what was going on from a molecular biology standpoint. It's easy to confuse those two things, right? We've got a partner, 23andMe. We're their sort of exclusive fertility partner. We're analyzing their data around infertility. And in doing so, we've got access to about 2 million total people. Let's say 1 million women. So access to 1 million women, remember we said somewhat 20% of them are going to have fertility problems. So 200,000 women with fertility problems, we have a genotype. Remember that 1,000, every 1,000 it differs? 23andMe has a technique for sampling the genome. It's called genotyping. For each person in there, they don't do a complete genome sequence the way we're just talking about. They just go look at a million, 1.2 million sites through, and then they use statistical association to kind of impute what's in between. Right? It's a neat trick. It's a lot cheaper than, I mean, just to be concrete, like it probably costs them about 30 or 40 bucks to do that genotype. Like I said, it costs about $2,000 to do a whole genome. That's why they do that. That said, you can do something called fine scale mapping, where you then say, hey, we've got this variant that statistically associates with this outcome. Now we're going to go look in that gene, and we're going to go figure out exactly what's going on, right? Because remember, 2 of these variants that are close together will stay in what's called linkage disequilibrium in many cases. They tend not to come apart because DNA doesn't get broken between them ever. The closer they are together, the less likely that's going to happen. When you and your brother share DNA, that brother's DNA and your DNA have been kind of mixed and matched from your parents by what's called crossover events. They just get less likely as they get closer together, obviously. So for us to then go, hey, that all that data from 23andMe, so like 200,000 women worth of like 1.2 million genotypes each, the data from the whole genome sequencing, so that's actually an enormous amount of data, because you've got all the 3.2 times 10 to the 9th bases for each of those patients, plus all the, what we call the sequencing metadata, tells us how we got it, and all the chemical information. So that's actually quite big. Just that data alone, to be clear, and you'll see this at the very end. So where do we put all that, right? Oh, this is terrible. You guys see this better? They can see it better on the screen. The point is that there's a great degree of heterogeneity in the data that we have, right? And this is a question that came up earlier. It's like asking, it's like, oh, you have electronic medical records data, and you have genome data. Remember, we've got, the genetic data here isn't just whole genome data. It's whole genome data, genotyping data, targeted genome data, blah, 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 from a bunch of different sources. That's got to be harmonized. Electronic medical records data from our own research, from the NIH and others, but also all the annotation work, right? You saw all that annotation work that we've been doing, manually annotating these things. So that's taking unstructured data from a variety of sources, including, in fact, medical records, not electronic ones, and then putting that in the system. And then finally, you don't see anything here, but it's you're also looking at the biological annotations, right? The other thing that's been going on, I think probably anybody who's in this field recognizes it, people have been building computer systems and tools and databases for holding biological information for a very long time. And we have all kinds of things, and kind of relevant to the immediately obvious is Jackson Labs built a catalog of mouse reproductive genes. Now, all the genes that they catalog at Jackson Labs, and they do a lot of work with mice, that can be shown to affect a mouse's reproduction. So a lot of mouse genetics. Why is that interesting? Well, mice and humans are quite different in theory, but they're both mammals, right? And in fact, share most of the same reproductive biology hardware. So for us to then say, hey, if we've got in the database a careful accounting of the genes that affect mouse reproduction, we can do experiments in the sense to say, hey, do those genes that affect mouse reproduction recur in the genes that we've got and predict patient failings that are similar to those in the mouse model? We do that by statistics, right? Yeah, we'll talk about that. So those are interesting tools, right? They're made to generate hypotheses that can be answered statistically and quantitatively, right? We talked again a little earlier about the p-values, right? Why do we care about these things? We're doing the math around these things to be able to carry out experiments in a way that is structured and formally answered. We're doing that by taking advantage of the data we're getting out of the genetics and the medical evidence data. So for this crowd, I think we're closing in on things that are of genuine interest, right? How is this stuff actually structured? And I'm going to go down through sort of 3 layers of discussion about it. This is at the highest layer, right? That says, A, there's a lot of external data here. It's HL7, SNOMED. This is data that has to do with how the data is structured, right? Because we leverage and take advantage of formal structures for this data that are generated outside. So Celmatix doesn't want to go invent how to structure medical data. We've got SNOMED, we've got HL7, we've got our Epsilon, but they're tools for this. We've also got a lot of, and this I sort of wrote this, you can't really read it here, but it's like semantic harmonization also requires that experts who sort of understand these terms and terminologies make sure that how we're mapping the data we're getting from our community is mapping in a proper way from a semantic standpoint. Remember I said the challenge is going out to the world and finding a whole bunch of different ways to represent the same data, and then building machinery that can harmonize that such that it can be queried in toto all together. And again, the stethoscope there, there are medical people, there are technical people who are working on that. In a similar way, remember we're talking about genetics, you've got the various different ways you can get that genetic and genomic information. Like I mentioned, 23andMe is a partner, they're doing genotyping, we're doing a couple of different flavors of whole genome sequencing. That too needs to be harmonized together in a single place. Now I will say, currently the genetic data, just the genetic data, is being stored in a very thin, very tall NoSQL data system. And that very thin, very tall NoSQL data system currently has, I think, just over 1 trillion records, right? So it's a very big system. Now those records are really small. I said this to somebody, I said, yeah, we're thinking about 4.25 trillion records. Oh my God, how do you manage that? Well, as I'm sure everybody in this audience recognizes, it's a very challenging indexing problem because it's so thin and so tall, and you're going to index these things. You've got to do so in a way that you can A, index them efficiently, and B, be able to query them efficiently, and be able to yield valuable results, right? And I'll walk through kind of explicitly how that's done in a minute in a subsequent slide, but just to say managing that, the bicameral, I think call this the bicameral mind, right? Here's the genetic part of the mind, here's the clinical part of the mind, being fed by systems, and then saying, hey, you've got to be able to query and analyze that. We've really leveraged, again, you'll see this, so we use some very standard tools for sort of querying and tracking that. But we're taking advantage of the fact that the standard tools have evolved incredibly fast over the last few years. So we're leveraging and taking advantage of the tools that are created in places like LinkedIn, and Twitter, and other places that are built for dealing with very large volumes of data in an efficient way. The kind of curious thing from my perspective, and somewhat surprising, is these tools are really underutilized in my field. People in, essentially what we call medical informatics and bioinformatics, only in a very limited way use some of these tools. And it probably comes back to sort of a lot of computer science or math people, right? These tools have evolved largely in the data science community. They've evolved with people who are very mathy. They're frankly very mathy tools, not easily used. So just to dive down a layer, just to sort of wrap it up, I'm going to hit just the architecture overview, and I'm going to re-show the slide a bunch of times as parts faded out, but I wanted you to see the whole thing in one piece. And again, remember the bicameral mind? Here's the clinical part of that. We'll walk through it. Here's the genome part of it. Again, look at this big, tall, thin thing. And then this is kind of, hey, how do people access that, right? This is the architecture overview. And again, we'll walk through each piece, but just kind of keep this as the summary architecture in mind. We'll walk through it. First question. Well, there's a lot of genomic data, right? 4.2 trillion records, 4.4 trillion records is a lot. This is, as I said, it's constructed here in a tool. We use Mongo for it. It could be anything, right? I mean, we can use Vertica. It could almost anything that sort of is really efficient for doing a tall, thin store. The reason we chose it the way we did is it allows us to support us queries across. VCF is just the name of the record, the data record that's used in genomics. For a million of I get almost 700 that are in there now, full exome samples. The total query time, by the way, of that tall thing, again, as I said, stored in Mongo, it's 1.5 trillion is now 4.25 trillion. The reason that this is fast is it's heavily sharded. Right? We're taking advantage of a biological fact. Remember very early when I showed you how these chromosomes are where the DNA is stored? Treat each of those chromosomes, right, at 23, thank you, 23andMe. Each of those chromosomes is one long string. It's technically actually two long strings joined in the middle by the centromere, but it doesn't matter. Imagine it's one long string with an address on it. That means that in theory, we could break this into 23 shards and never cross one of those shards, ever. Every query would absolutely only go to the one shard, right? The problem is the chromosomes vary vastly in length. Chromosome 1 is really big, and chromosome 20, 21, 22, 23 are all really small. So how you balance them, we actually break it into 5 shards. One shard contains one set of chromosomes, and we balance them so that the shards are roughly the same size. We actually believe we'll probably end up sharding them more, we just haven't done it. But just to give you an example, querying the current version, which is about 4.25 trillion, the query only takes about 186 milliseconds, right? Why it does that is we went and pre-indexed this thing against that address, remember that location in its chromosome. So there's really just 2 steps, which chromosome are you in? And where is it? Go get it. Now get you that base, and get you that base and any number of patients. You can see why this isn't a very slow operation. It's quick. And it's also why it doesn't really matter what we use as a database. We could have used almost anything. We just chose something free. And then sharding, hey, this is what I mean about distributing those pieces between the shards, right? Loading that data is entirely parallel. Now, loading pieces really, really, really big. We'll talk about it in a minute. We take advantage of sort of elastic compute to allow us to expand the size of the machines and then decrease them. So loading is a very compute-intensive process, but querying isn't. So we load on a really big machine and we query a much smaller one. There is more operational overhead when you're dealing with shards. I mean, I think all of you probably know this. If you're dealing with parallel databases, it gets a little tricky. At the very end, I'll sort of mention this. And I said it's sort of interesting. It's flexible and expensive. If people want to come up and talk to me afterwards, we've actually done some pretty interesting things as we've kind of gone from a pure AWS deployment over the last year to a hybrid cloud deployment. It's just been interesting that we sort of did everything in Amazon. As we've been running it, and we're seeing the operational costs we're gradually pulling more and more back, which has frankly surprised me. Clinical data, right? Again, the data that's coming from all of these clinics, right? So we're plugged into these clinics' electronic medical record systems. We're lucky, again, and we're really doing direct database level access. Anybody who's sort of in this business will go, what, do you get an HL7 feed from one of these guys? How does that work? Since these are all largely pretty bespoke systems, we just directly hit the database. In doing so, the tool that we're using is we use Kafka. This is the Apache Kafka project. It's a way for us to do sort of manage streams. The nice thing about this is we can build streams. I have a set of bullets I'll show you. The advantage here is we can then build a stream for each of these clinics. We can stream the data as documents for those clinics. We have individual stream workers, which do things like anonymize and de-identify the data, that harmonize it. One of the beauties of Kafka is it's got actually a really nice schema system. So that schema system can represent the kinds of data in its harmonized form. And then the harmonization is just a bunch of stream workers that refute a bunch of business rules that we use to do the transformation. And that's all sort of automated, right? People came up with it. We've sort of exposed it. I mean, for people who care, we've kind of exposed it in a Confluent. It's literally like Atlassian Confluence. But the underlying code and the code layer it runs is just running its stream workers in the background. What that means is that we can harmonize and converge the data from all these places and be assured that it's secure, that it's de-identified, it's anonymized, and that it's all in one universal format for which there's a standard schema. Right? The database where it lands, I mean, you know how these things work, there's a sink, right? And so the streaming analytics is just running the analytics as a stream. We can do all kinds of on-the-fly analytics that we do to kind of track what's going on, but mostly, frankly, those standardized data elements, once they're converged, they just go on a Postgres database, right? This is a good case for relational. And again, the clinical data is in 4.25 trillion records. This is what I don't know, it's 670,000, I think we're at about 7.5 million unique data points over a billion records, right? So it's not crazy. The nice thing is it is kind of a relational database. It's a harmonized, standardized relational database. And then it's updated in real time. Like, as these things change, we can take advantage of the streaming system. We can update it. We can play it back through the streams if we want to. But primarily, we just do most of our querying against the data in the database. I mentioned the Kafka. I won't go into detail, but the schema registry from Kafka is essentially what allows us to sort of manage and handle the schema, the representative schema, going through those streams in a way that's isolated from the systems themselves. We don't have to go mess with and deal with the ugly ways that these guys have built their EMR systems because they are uniformly terrible. Every one of them is terrible. One of the things we talk about a lot is, hey, we should get in the EMR business. Those are really terrible. We'd be much better at it. And then we think a minute, we go, no, we're not going to get out of business. That would be really terrible. The upside is that Kafka builds in encryption and stream for us to handle electronic medical records data, to comply with the agreements, the business associates agreement we have to comply with. This is HIPAA, remember? In order to do that, we have to guarantee that we encrypt data at rest and in transmission. Well, Kafka will take care of encrypting and transmission. That's cool. At rest, well, we say we had to roll around. We obviously leverage Postgres for that. So, Conways Law, right? Organizations which design systems are constrained to produce designs which are copies of the communication structures, right? This is not, we are extremely collaborative. We're a standard startup like this, nobody has any offices. So it's interesting that everybody's got their own thing going. We're all communicating all the time. It was a little funny that we followed Conway's Law exactly, building systems that'll let that kind of complex group of kind of constantly interacting people kind of all do their own thing at the same time and still yield something valuable. So, again, as you might expect, this is getting big enough to where we do materialize some views in order to answer the kinds of questions people answer all the time, and to feed those analytics and AI programs I was talking about before. Finally, hey, how do you make it cost reasonable? Well, as I completely whited out, AWS and on-premise, we were running quite a bit. We figured it was going to be about 1.2 million a year to store a year's worth of samples. Remember, these are really fairly big and complex. We ended up building on-site hardware in order to enable us to do it. It's just one of these things where it curiously turned out to be cheaper for us to do it on-site than not. There are hidden costs in that, of course, right? And I'm always cautious about doing it. But as I always say, at the end of the day, we just look and see what we actually spent. And our actual spend this year, in 2017, was less than our previous year, having done it in AWS. So we actually spent less and did more. So lastly, we use Kubernetes for managing it. This is all containerized infrastructure, for automated deployment. Again, like I said, we feel really lucky that folks like LinkedIn, Twitter, and these other guys have had these same problems. How do we manage these flotillas of computers doing a lot of complicated things, need to be done fast in a very parallel way? And we turn to the same sort of open source and readily available tools, containerization, Kubernetes, and so on to do it. And then, as I said, we're kind of cloud bursting. We've gone to a hybrid infrastructure where the majority of the compute and the storage is done. Anything needs to be running at 80 to 90% all the time is just run on-prem, and then the remainder is burst out to the cloud when we need more compute. Won't talk too much about that. I mean, this is just as you might expect. And then finally, how do we allow the scientists? We use Pachyderm for this. Pachyderm is a really interesting approach because what it is essentially Dockerization of Pachyderm essentially provides, as we say, like Git for data science. It provides a version control, fully version repository that is intrinsically able to sort of feed into the container in a parallel way. And again, I won't talk too much about it, but it's as easy inside the containers as just reading to a named directory. Pachyderm is taking care of then spraying that out through the parallel set of information that's needed. So this has been great because we've been able to give these sort of Pachyderm containers to our data scientists and to our bioinformatics people who are not, by and large, sort of hardcore software engineers. And they're able to take off and just run and build fully parallelized compute infrastructure that we can run either locally or in the cloud as we see it, leave it up to DevOps. So where does this all go? Well, like our products are driving better outcomes for fertility. It's gotten really great. I want to get a slide in here. We've had our first fertilome babies. So we've had our first people who were unable to conceive before. We're able to identify what was going wrong, intervene, and have kids. This to me is really exciting. Like we're able to help people. And I think as I mentioned before, especially with sort of the way millennials are treating this is going to become more and more important. The products become clarity and personalization to the fertility process. This is what it looks like. Like how amazing is this, right? Like, oh, just collect some DNA and we're going to give you a really important answer that's really useful. And the future, we believe, is personalized medicine, and not just in women's health and infertility, where I work, but across the board. And I want to make sure I acknowledge, like, I wish this were all my work, and like I just did it. I didn't. There's a group of really enormously talented people down on Wall Street who are doing amazing, amazing, crazy work. And I want to make sure that they get credit for it. And that's it. I'll take questions if people are interested. Is there a microphone? Oh, okay. Yes.
I have a question about very interesting. This is kind of like your use of hybrid cloud.
Yeah.
You found that on premise. It's almost like a man bites dog story. How did you find that basically that running on premises ended up being cheaper than in cloud?
It's crazy, right? I mean, it's one of these things that sounds absolutely improbable. So I'm going to give you a simple answer, right? The simple answer is, at a certain threshold, you're going to make slightly different trade-offs than they're making in the cloud infrastructure, right? So if you make an assumption that you're going to run the thing 100.0% of the time, like that computer is turned on and running all the time, then you've made certain trade-offs, and they've made certain trade-offs. They've got to build in more robustness, more redundancy, more, in order to be able to guarantee you, the customer, can get 100% from that thing all the time. We, on the other hand, are monitoring this thing carefully. And I always like to say, we know what shortcuts we're taking. We're taking shortcuts we can live with, which our cloud provider might not. I will say, in practice, this has enabled us to cut our costs about in half for things that are running at least 80%. Right? Now, that said, here's some huge caveats. Caveat number one, we're running at the absolute fringe edge of what's available in terms of compute hardware from cloud providers. In fact, up until a month ago, Google couldn't provide us in their cloud what we needed from a compute standpoint. We needed across a very, as you saw, very high memory machines to do the genomics and very fast machines to do it, fast. And Google was sort of one or the other. Amazon kind of has a thing up here that's both, it's really expensive. So A, we're living at a very expensive point. B, we're essentially taking advantage of the fact that we can build to support that very edge use case. Tons of memory, tons of processors, really fast running in Equinix and Secaucus. Equinix and Secaucus is a pretty cheap place to run compute. We get good deals from Dell because they like us. And they don't require a lot of management. I mean, we really have very little, I technically only have 2 IT people that represent our DevOps, our IT guy, they're doing everything. We also have a couple of desktop support people, but just really the people running all this is very small. The reason we're able to do it is, B, we can use that same compute to run all of this other crap we're running all the time containerized. So we have Docker containers running all the time. We use Kubernetes to manage it. We've got a good DevOps team. And they're watching, I love their desks. They got these big monitors that have all the monitors of everything that's going on. But we will then unload, we'll contain the Kubernetes Docker fleet to just unload a lot of the work that's going on and store it temporarily. And then we run these big analyses up and use the thing like it's a supercomputer. But the advantage of running a fully Dockerized infrastructure is that within a second, within a minute, we could have completely unloaded all those supercomputer jobs, and that thing's now not a supercomputer, it's a whole bunch of fully virtualized instances that are doing all the little bits and bobs that you do to run a business. So we're kind of running our own cloud there, and since we're running it at full tilt, we just wouldn't be able to reasonably do that. And then I say, people say, well, is that really true? I say, yeah, I mean, I can just say, I know exactly what I paid, and we were able to shave off quite a bit.
But this is just kind of this is kind of.
Hang on.
Can I?
You can answer it.
Okay. This is just kind of a general case because Amazon and Google, in business to make money. So if on-premise would not be cheaper than in cloud, then they will not make money. So if you know what you're doing, on-premise, even in general case, will always be cheaper.
And you have the right people.
Yeah, I think this is
Yeah,
where,
and you have the right people. That's what I said. If you know what you're doing, on-premise will always be cheaper. Otherwise, Amazon and Google will go out of business.
You're talking about some very
So
ideal
why is that even the question? Otherwise,
I'm just saying, you're talking about some very ideal, extreme, extreme end of the envelope circumstance that's probably not typical for most organizations. But you're basically running some very extreme use cases.
no, but this is the same thing. Like you go to supermarket and you get like a piece of meat at certain price. If you go to restaurant, you get it for 10 times more. Not 10% more, but 10 times more. And you're still happy because somebody else, grilling it for you. Right? The same thing here. They have so big margin that no matter what, they will make money.
sorry, go ahead.
I have a question on your data for the outcomes. You said that awesome babies have been conceived with your recommendations. Do you have data on how different those recommendations were from what standard medical practice in infertility is?
How about we're in the process of conducting a large-scale trial right now where I can give you statistical data. This is still early days. Just to be clear, we received our New York State approval to sell this test generally last week. Yay! But we did quite a bit, obviously, of preparatory work, and a number of academic physicians have been working with it for about a year, right? So we're working on I can answer your question in quantitative, Mark would be comfortable detail, probably in another year. That said, let me answer about these kids, as I know what happened exactly, anecdotal, if that's okay, right? And the anecdotal answer is this. I'll give you an example. If you look in those fertilome genes that we're talking about, several of the important genes we're looking at are genes in the immune system. It turns out that certain autoimmune disorders, or things that are closely related to autoimmune disorders, can actually cause complications of pregnancy, cause complications of implantation, and actually can cause things like premature ovarian failure and so on, right? Just not surprisingly, this isn't just infertility. Like pretty much every type of disease has some immune complication, right? Not surprising. However, it turns out that in infertility treatment, it's known that, but considered kind of a fringe treatment, to do some treatment that would look a little bit like cellular immunity against transplant or rheumatoid arthritis, these other autoimmune diseases. Things you use to treat an autoimmune disease, you can treat pregnancy. Not surprisingly, this is not widely done. These are drugs that are tough, they can be a mess. It's like, you know how it is with steroids, imagine that 10 times worse. So it's not something you want to give to a woman who wants to conceive routinely. So they generally don't. But it's known in the literature that occasionally these treatments work. And in fact, there's sort of a little cottage industry south of the border for women who want to get pregnant. They're just failing and failing. Like, hey, come down here. We'll do this treatment, and it's kind of crazy. But it does sometimes work. That's the crazy thing. So if you go to a fertility meeting, I was at ASRM, the fertility meeting. They have the sperm bite there. It's crazy, right? Where that debate rages. Is that a good thing to do or not? In several of the cases that I'm talking about here, they had women who, when they did the fertilome test, like, wait a minute, they've got a flotilla of these diseases that are associated with the markers that are associated with immune diseases. This woman, Amber Cooper at WashU in St. Louis, said, okay, great, what if I take these women and actually treat them with one of these not frequently done things? What will happen? In of the cases that she tried, 4 of the 5 cases that she tried, and she did this. I would not, she said, I just never did this treatment. I don't believe it or not, I'm going to try it. Those women were pregnant. Like, we have an actual baby, we have 2 babies born now from that. So, there's a case where there is a known treatment, an intervention that you can do, that the physician looked at this and they said, you know what, this looks like a candidate for a treatment that I don't normally prescribe. I'm going to prescribe it. Now you might say, why don't they just prescribe that for everybody? It's not a great treatment. It generally doesn't work. And it's in fact generally worse than it is better because these treatments are pretty rough, right? So I can give you other examples of recurrent implantation and so on, but all of these things basically point and say do something a little different than you would normally do. That's just the most extreme case where there are people, like it's in debate. We believe that as we're narrowing this down, I didn't really talk about this at all. One of the real reasons we're in business here isn't just to sort of have this for a lung test that can, help people conceive. We're also looking to say, hey, what are the actual variants? Remember I talked about fine scale mapping, like what's actually going on in molecular biology there? We've got 2 pharmaceuticals deals where we'll probably do another this year, where we're literally looking to say, can we identify drug candidates? Probably not in pregnancy, but probably more in endometriosis and related disorders, to say, let's point the drug companies that we work with at potential treatments for these things and let them go build drugs. We're not going to be a pharmaceutical company. But that's the other outcome, is we're starting to collect all of this data and all this genetic data, and we've got all these women who've agreed to research consent to let us do sequencing on them. And we can then go find things that we can turn into unique intellectual property. Like I would say, Celmatix is a biotech company, right? That's really where we're going to end up. Think of the sort of Furlome and the Polaris stuff as a way for us to sort of get from here to there, right? Does that answer your question? Yeah. We'll have more when we publish our results, right?
I've seen that you work with hedge funds, so what's something that you have contributed with?
The question is, I see you work with hedge funds. What did you? I worked at Bridgewater Associates. I led the data collaboration team, which is the in-house data science team there. I have to be very careful what I say because I'm under non-disclosure. I helped leverage natural language processing and artificial intelligence techniques, machine learning techniques, to help analyze written data, like just written information, and to yield structured data that can be used for a variety of business decisions. And that's the answer I can give you. I'm sorry I can't tell you more. It was really fun, and it's a very crazy place.
Sure. Now, I guess just with your fertility assay and your system architecture for looking at all this data, are you able to do point-of-care diagnostics where somebody shows up to the physician's office and say, hey, they need an answer, like, sort of like...
It's literally... I was literally in the process before I came over here of taking apart a small micro reader that does I think it's an ELISA assay. At any rate, it's a very small illuminated assay. Because the idea that we could do a chip read on a very small number of variants at the point of care of the doctor's office. We may literally do this. We'll probably do it with licensure. This is why I had that machine. They gave it to me. We probably wouldn't build the systems ourselves. We would license it.
There's a prototype already that
Yeah, there's a machine that when I was at Booz Allen, one of the things we did with DARPA was build a system that was for identifying biological warfare agents in theater. So they were for soldiers to be able to use carry on their belt. I reached back out to those guys and said, hey, you built that really neat thing for doing biological warfare. Could we use it for fertility? They were a little surprised. And they're like, okay. But there are a lot of these things, right? So I even point you guys, if you want something really amusing, go look up and I think it's called the Minion, M-I-N-I-O-N. It is a sequencer that literally plugs into your iPhone, does DNA sequencing. So we're so close to that. This is just a question of like who's going to make the first of these machines that's readily available in a physician's office. But we won't make it. We'll license.
Sure.
Yes.
Curious, either in the literature or in your the Furlome assays and that sort of thing, is there sort of a bias towards exome DNA and sort of the either express sequences or the conventional promoters and that sort of thing?
This is a great ad. The question is like
to all the other stuff that is probably really important.
This is something, the question is, hey, the variants that you guys are using in Furlome, or the things that you found in your research, primarily in the genes themselves, or in the expressed part of the genes, remember the bits that I said got turned into protein, or are they in the other 99% of the genome, right? We used to call that junk DNA when I was in grad school. That's probably really not true. Like probably that just as DNA, we don't know what it does. It's sort of supporting that. The majority of the variants that we found in literature search, these are markers, remember, by and large, they aren't necessarily functional, are in the exome. With a smattering, in intronic regions, and some that are actually relatively distant from any genes that we know of. We arbitrarily give it a gene name because we pick the closest one, but it ain't anywhere near it. Not close enough to be a promoter or enhancer, any of the machinery that we know of. So I think the proper answer to your question is, were I to guess, my guess is most of these are markers for actual functional variants. So something that makes a change to the protein that causes it not to work properly. A, I'm old school, C, gray hair. B, it's just I kind of have a guess as to what some of them are already. That said, there are a handful of these things that I think are probably structural. They just have to do with how the DNA folds. But we don't really know. Like, we just don't know. And I mean, it's unfortunate because there's so much we don't know.
Or much more complicated.
Yeah, it's probably more complicated than I think.
Yeah.
Last question.
Okay, last question.
I was wondering how is going about giving out the fertility kits? Is it direct to consumer or to clinics? And what's the price point right now?
This is a great question. Like, how are you getting those things out? Right now, they're going to physicians, right? In fact, even more specifically, they're going to IVF clinics. There's quite a bit of training we have to do to teach people how to use this thing. Not as much the kid. I mean, the kid is like, draw blood, spit, send in. Like, that's easy. There's quite a bit of training to help the doctors interpret it. So as a result, we've been pretty focused, right? That said, within our 2018 plan is the estimation that we will be rolling out a direct-to-consumer product in late 2018, early 2020. Again, we've been validating saliva for that reason. That's why we made a saliva kit. And we're looking at, again, channel partners that would help us do it. Again, we're not going to put all the kits in Walmart or whatever, right? And just to answer your question explicitly, we literally make all of the kits in our laboratory. We have a laboratory in Brooklyn in the Biotech Incubator City downstate. We have a laboratory in New Jersey up by Rutgers. We literally make the kits there. Because we're doing, for the reason that I mentioned before, hey, we just got approval, so we were doing huge volume. You know, we're doing thousands right now. We're going to shift up to tens of thousands. Allied will probably build them for us. Like, we've made a deal with them, we just haven't started yet.
That is all the time we have tonight, but Dr. Mark Adams is here for one-on-one questions. Feel free to come